Hi, everybody. We're going to be talking overall about parts of cells. What I would like to do is discuss some of the first parts of cells. We'll talk about up to bacteria, talk a little bit about microscopes, and we'll then talk about how these things kind of come together to see the different types of cells that we need to for class. So in chapter 8. So we really need to end with prokaryotes, eukaryotes, and the differences between those two. So, with that, who discovered cells? Robert Hooke is given credit for um, for coining the term cell. So what he did is he had a compound light microscope, and what he did is he was able to look at cork cells, a small sample of cork cells underneath his microscope, and he goes, they look like how monks kind of live, the rooms that monks live, those are called cells. So he said they kind of look like cells. And what's interesting is that he was not looking at living cells when he coined this term. He was actually looking at dead cells, the remnants of cells. And that I think is kind of fascinating. He's not the only one who kind of used some of this stuff, but his name comes up within it. So microscopes. This is the way microscopes look. You will need to know parts of the microscope, and you can always pause this and kind of look at the different parts. Functions of these things we should know. We're going to be doing this more virtual with our labs. Within it, so these are the objectives. So it's a compound light microscope. So there's objective lenses, okay, and we can kind of sort through those. But there's also another lens, the eyepiece, and that's why it's called a compound light microscope, is because it's using two of these lenses at the same time, and that gives enough magnifying effect. And so the total magnification is always the product. That's right. You multiply the product of the two magnifications of the lenses. So if like your eyepiece is say 5 and your objective lens is 5, then it's a what? 25 times. So we're actually, the product of the two. We'll talk about that a little bit more in our lab. So what's more interesting is what we can see with them. Until the microscope, really, people had no idea what a cell was. They didn't know what things were made out of, and I'm talking about living things, obviously. So the light microscope really opened up a new understanding of what was going on. And so we'll talk about some of the in class. We'll talk about some of the major contributors to kind of identifying what a cell is and who they were. Another type of microscope that aided our further understanding of things is the electron microscope, and that gives us a micrograph, an electron micrograph of different things. So again, now we're able to see parts of cells more clearly. We can scale down to what a virus is, and then even small parts of cells, proteins and things like that. But again, we're not as worried about the atomic level. So compound, they're both very useful. Compound light microscope is able to see cells very clearly. You can see cells that are living versus an electron microscope. Electron microscope is you would have to kill, they say fix the specimen, and then you would be able to see them. But again, look at the resolution though on an electron microscope. This is actually a scanning electron microscope. Beautiful pictures, right, within these things. These are pollen grains. Whereas the Elodia, right, again, not as clear, but very effective, because now we're talking about a living organism versus something that we had to fix and kill. We study biology, which is living things, so we're going to have to kind of make some assumptions with how these things interact at these higher magnifications, because you can't see them living. Cost $50,000. A good one, probably about $500 for a compound light microscope. So obviously in class, we'd be using the compound much more because it's practical for us. Cell theory. So these scientists came up with basically three principles of cells and what cells do. And so over and over, they kind of refine this idea. So cell theory is really, what do we understand about cells? What's well tested? What's well accepted? Well, all things that are alive are made out of them. Cells will only come from other cells. And basically cells are the structural and organizational unit of living things. Okay, and so the question then becomes, well, what about viruses? Well, we talked about that a little bit already within it. So another question that comes up, which is interesting, is, well, where did the first cell come from? And so we will talk a little bit about that with evolution and biochemical evolution. And there's some evidence for some plausible ideas, and whether they're testable or not becomes kind of an issue with this. Okay. So, what is the first separation of types of cells? One is having to do with the nucleus. So, prokaryotic cells or bacterial cells do not have a nucleus. Okay, now obviously, scale wise, these are not at the same scale. You have to kind of look at this from a perspective. Bacterial cells are much, much smaller than eukaryotic cells. Okay, so realistically, you're talking about hundreds of trillions on a pinhead. So notice they're actually using an electron microscope to see this, okay, versus a eukaryotic cell, which we would only need a compound light microscope to see the whole thing. So bacteria are like on the very verge of being seen with the compound light microscope within this. So this is a Bacilla. E. coli is the bacterium that lives in our guts. Some have flagella. They have a couple of cell walls, okay, within this. The cell wall, though, is made of a different structure called peptidoglycan. They do have DNA within them, but notice there's no surrounding membrane around that. They do have ribosomes that make proteins, and then they have some other structures on the outside. Sometimes they can become toxic. They're called fimbriae, these little protein extensions. They might have a protein coat around the outside. They call that a capsule within it. Now, eukaryotic cells do have a nucleus. Okay, so they have a membrane that surrounds the DNA, and there's another structure, a darker stained structure in here called the nucleolus. Now, I purposely put a plant cell, a plant picture with an animal cell, and the reason why I did that is because both plant and animal cells are eukaryotic. Now, one type of cell that we don't have here is like a fungi. Okay, so fungi are also eukaryotic cells, and there's a lot of different single-celled microorganisms called protists that are eukaryotic as well. Okay, but so from this standpoint, you can see that there's a lot of different types. There's some different types of groups of eukaryotic cells that might not be represented right here, right now. Okay, with that, there are a vast majority of diversity of bacteria as well. Okay, within it. So if we want to talk specifically about bacteria, no nuclei, they're 10 times smaller than eukaryotes, so much smaller. Okay, again, an electron micrograph, they have a capsule, which is a protein coat that surrounds it. It kind of glues, allows it to glue to other cells. They have a cell wall. Okay, inside of that, there's a cell membrane, and they call that a plasma membrane inside of that. The ribosomes are used to make proteins, and then they have a nucleoid. This is just the area where they find the DNA. Okay, now that's about it. So again, cell wall is used for protection, made of peptidoglycan. They have a cell membrane that's really for regulation. I don't know why I put protection twice. Nucleoid region, obviously where the DNA is found. They call that a chromosome, just a single circular form of that. And then ribosomes that help make proteins. Now, eukaryotic cells can take on a number of different shapes, but they have some similarities that we want to talk about later. So one major division between those is plant and animal cells. But again, I want to talk about eukaryotics later. So I hope this helps kind of organize information in your brain, and I'll talk to you soon.